Hello, hello, hello. Okay, so anybody have any questions about the trip operon before we get going on eukaryotic gene expression?
No, but I have a question about that extra credit.
Sure.
Do you need us to send you that confirmation email or anything like that? Okay.
No, there's some website that I can check completion rates on, and I'd rather do it that way too, because that way it's completely anonymous who has not commented.
Okay.
Yeah. So this again, this is an exercise in giving you like a little bit of extra help, but also giving me a useful sample size when it comes to feedback and evaluation. So it's not intended as a bribe or an incentive to say nice things. It's just simply an incentive to do it, and that's it. Okay, so dang it, where'd chat be? Chat be here. Well, it ain't over until the grades are submitted, Robert. So gotta keep fighting. And every bit of work that you do on this course will help you with subsequent courses too. So it's not just an effort solely for this one. It's also for all of them. Well, not all of them. I mean, if you're doing kind of one of the fluffy panda type courses, then maybe this wouldn't help you much. But hopefully that's not going to be the only thing that you do here. Okay. So now we need to get onto eukaryotic gene regulation. So remember, in bacteria, regulation of genes involved in the same processes is coordinated simply by grouping those genes together into operons. Right. In eukaryotes, that we still have a need for coordinate or coordinated gene expression, but we don't do it by grouping them together. We do it through yeah, giant pandas. Can't stand them. Any chance I get, Robert, any chance. Instead, eukaryotes coordinate gene expression through the proteins that bind to and activate or repress expression. I'm talking about transcription at least. And there are lots and lots of levels to this, right? So in some ways, prokaryotic gene expression or control of gene expression is more complex in like the mechanisms, at least at the level that we look at it at. But in general, eukaryotes have far, far more layers, right? Multiple layers of how the production of a protein from a gene is controlled, right? And so these are laid out quite neatly, and most likely we're going to be taking this into lecture on Tuesday. I don't think we'll get through this today because there's a lot of stuff to talk about. God, what are those dogs doing? Sorry, they're very distracting. They're kind of just tearing their asses around the house, chewing at each other, or small ones chewing at the big one, basically. So we've got a series of scales and processes, right? And so what I want you to get is that each individual cell, oh no, it'll be on the exam, definitely. Everything up until the end of our last class together will be valid material for the exam. Or one thing to remind you too, though, is the exam the last exam, again, it's not the final, right? Exam 4 opens on Wednesday.
So do we have exam 4 and then a final, or just exam 4 is our last exam?
Just exam 4.
Okay.
What day is Wednesday next week? It's the 28th. Oh, thank you, Adam. You were quicker than I was. Yeah, so I want to just double check, see what's in there. Yeah, so that exam will be open from 9 a.m. to 11:59 p.m. Wednesday 28th. So a little bit different here. We're not going to be doing it over kind of 2 days, 1.5 days, but it'll be open earlier on the day that it will open. So you will have like the whole day to fit that into your schedule to be able to take that exam. Yeah, so Adam, typically, sometimes things take different amounts of time to work through in terms of subject matter. So control of gene expression in prokaryotes and eukaryotes is like the main chunk of this last segment of the course. And so if we get time, then it'll be cool to be able to go into something like the biotechnology and genetic modification stuff, which I like to do at the end of the course. But if not, that stuff is like, that's the icing and the cherry on top of the cake. I need to make sure you get the cake, right? And so that's why there's always a little bit of kind of slack time at the end of the semester, just to be able to make sure we cover everything we need to. But hopefully there'll still be time for that stuff, because it is really cool to talk about. Really enjoy talking about that stuff. Okay, so we have what's the context, right? The context is we have a cell. That cell is one of many cells. We talk about multicellular organisms here, although obviously there are unicellular eukaryotes, yeast and various parasites and stuff. But in terms of eukaryotic cell, you're going to receive a signal, some kind of information from outside of the cell. Now, in terms of scales, we have many different scales at which that information is going to be processed, right, in order to result in a change in gene expression, right? So remember, these cells, either as part of a larger organism or kind of wandering around on their own, need to have a way of turning on or off gene expression, right, to control what genes are expressed in response to what. In response to what signals and stimuli and information. Dogs, will you just go somewhere else? Oh, Little Bit is trying to steal Big Bit's bone. Where is she? So funny. She'll be back again in a little bit. She's just tearing around a little bit before she loops back. I don't have these kinds of distractions when I'm lecturing in a lecture room, that's for sure. Yeah. Yeah, it's no day is ever the same, that's for sure. Anyway, so we can control gene expression in eukaryotes at the big scale level. And so genes are typically, they're involved in the same process, often are in similar regions of a chromosome, right? So we're able to open up or shut down individual regions and kind of basically turn on and off programs of gene expression as a result. Also, individual genes, we can change the state of chromatin for fairly fine scale control of gene expression of individual genes. Right, so before we even get talking about the control of transcription, we need to talk about how accessible that DNA is. Right, and so that's all about chromatin and chromatin status, if it's open or shut, if it's available for transcription or repressed. Yeah, I wouldn't want to try and step in Jim Carrey's shoes, Robert. Besides, he has bigger feet than I do, as far as I'm aware. Once that DNA is opened up, right, so it's available, now we have the control of transcription, right? And as you can see from this particular diagram, control of transcription is a much more involved process in eukaryotes, in the sense that there are many more factors involved, many more bits of DNA, many more proteins. It's generally much more tunable, I guess is probably the best way of putting it. So once we have transcribed that gene, well, now we're talking about RNA processing, right? So in eukaryotes, genes have introns, and we can actually control which introns and exons are removed. You can actually make more than one protein from one gene, which is a kind of a weird thing to think about, right? We've really been following the fairly straightforward idea of, you know, DNA gets transcribed into mRNA, which gets to make a protein, right? One gene, one protein kind of deal. Well, it's actually more complex than that. You can make multiple different proteins from the same gene. And I'll give you an example of a gene that I studied for a while, which has that kind of setup, which is pretty funky. I'll show you some pretty pictures. So far, we're just talking about turning on genes. Well, we also have the ability to turn them off or reduce transcription, or not transcription, reduce expression. And we can do that by controlling the stability of the mRNA. So all mRNAs are not equal in terms of how long they persist for. So they vary from gene to gene as to how long they hang around for. Hello again, Robert. Ah, making fresh coffee. I'm jealous, even though I've already had 2 cups. So what we call housekeeping genes, basically, in other words, things that are always needed, like ribosomes, let's say. Produce mRNA that persists for a very long time. For example, genes that are specific to a very particular circumstance, right? May produce mRNAs that exist for very little time. Because if you think about it, as long as an mRNA is around, it can be translated. So if you only want a very specific amount of protein, or you want to be able to control how long you're making that protein for, you not only have to control whether or not you turn the gene on, sorry, you also have to control how long the mRNA hangs around for. Because even if you turn a gene on for just a really short amount of time, if that mRNA persists, you'll still get protein produced from that mRNA. So if you only need a gene expressed for a very narrow window of development, let's say, I don't know, a couple of days during embryonic development, in order to, I don't know, get your like limb nubs developing, for example. Not only do you need to stop transcribing it, but you also have to get rid of the mRNA so you're not going to keep translating it. And so again, this is another kind of wrinkle of eukaryotes having transcription and translation at different times and in different places. They're not coupled the way they are in prokaryotes. Okay. Now, even when you do produce a protein, that protein requires a lot of work to be done to it. So the polypeptide, it needs to be folded into the correct conformation. Almost, well, not all proteins, but a lot of proteins require subsequent processing. You know, they get bits kind of cut off of them, they get stuff added to them, whether it's sugar moieties to make glycoproteins that you'll find on cell membranes, phosphate groups or other functional groups that change its activity. Right, so functional protein is not necessarily produced by translation. There's almost always subsequent steps that are required before that protein is active. And once it is active, then there are subsequent steps where maybe it needs transporting to the correct place. Great example of that is insulin, which is a protein. It's a endocrine hormone, basically. And insulin is secreted, right? So your beta cells in your pancreas kind of dump it out into your bloodstream whenever your blood sugar levels go up, because insulin promotes the absorption of glucose from your blood. It's kind of part of your glucose homeostasis. So the insulin peptide, the actual protein that does that job, has to be cleaved like several different places and then put in a situation where it can be exported or secreted from the cell on demand. Until that point, that insulin is not active. Doesn't matter how much you have in vesicles inside your beta cells, if it doesn't get into your bloodstream, it can't do its job. So really you have to think about protein function in a kind of a holistic way. It's not just like the ball of amino acids that does stuff, right? It's what gets done to that, where that protein ends up, what state it's in, whether it's turned on or turned off. You know, even after all of this, you can modify protein activity. Hey, so there are many steps between receiving that information and getting a functional protein out the end of it. And in addition to that, those proteins don't necessarily hang around forever. Those proteins can then get, you know, reabsorbed, broken down, removed from the picture. And so that signal up here might be received by a protein on the surface of the cell, a cell signaling receptor. Quite often, once that signal is received, that receptor gets destroyed. So if you want to keep receiving that signal, you have to keep transcribing and translating and modifying and exporting or directing that protein to where it needs to be. So you really need to consider the whole picture, right? It's not one individual step that necessarily determines whether or not you'll have functional protein. It's all of that kind of summed together in a sense. If you were to write a long equation, each of these steps would be a function in that equation to be able to figure out overall expression of that gene. So we're going to go through this. This PowerPoint's kind of organized to follow this, I don't know, flow diagram, I guess one would call it. And so we're going to start off talking about chromatin, and then we're going to work through each of these steps in turn. So by the time we're done, you're going to have a very complete picture of all of the different ways in which gene expression can be controlled in eukaryotes. And this is also a neat kind of thing to talk about, because if you look at these two cells, so these are confocal, laser confocal microscopy pictures of two different cell types. I think they're grown in vitro in cell culture by the looks of it. And so these are expressing two different fluorescent markers, or they're immunolabeled with two different antibodies. I'm not sure which. But either way, wherever you see green is where you see actin, which is a structural protein, as is microtubulin in red, which is more of a like a cellular highway type protein, essentially, also involved in cell division. So these two cells, I love looking at confocal pictures. It's just super fun. Actually, I'll show you a really neat picture. A little bit of a tangent. This is a picture I absolutely love showing to students. Look at that. This is a confocal image of an alligator embryo. Right, so yeah, I love showing cool pictures. So I love microscopy. In some senses, a microscopist as well. I don't do a lot of confocal work, but it's always fun to talk about as well. So this is basically a way of taking slices of an image throughout a three-dimensional kind of structure, right? So each slice is perfectly in focus. And then once you take all those slices, and you can have potentially hundreds of images, use software to stick them all into one image. So every part of that image is in focus, right? So this is a alligator embryo, you can see the heart is developing here, the myocardium. These blocks down here are what are called somites. They're blocks of tissue that will become vertebrae and muscle. And so essentially you have vertebrae form kind of through the division of these somites. And then the green stuff, as far as I'm aware, are neurons, right? So here's the developing embryo alligator brain, which looks pretty small, and the spinal column, which is going down here, and all the innervation of the different muscles and stuff, right? So that's just a cool picture. And it's also a really fun one to show. Now, what was I actually talking about? Here you go. So these are two types of cells, right? We've got neurons here, and you have keratinocytes, which are basically the cells that make up the either like your nails or your hair or something like that, right? And you can see just by looking at them, they have very different morphology, right? Neurons have a cell body, and then they have these processes that come out called neurites, either axons or dendrites. Those are used for signaling to each other. And this is a neuron that's busy growing. So you can see these growth cones are how these neurites basically kind of squinch their way to their destination to make those connections. Now you look at keratinocytes, you've got a very what's called squamous form. They're very flat, they're spread out. They almost look like plant cells, right? They've got these kind of tight junctions between each cell, right, to allow them to form this layer. And you've also got a very different distribution of these two proteins, right? In neurons, the actin's very much involved in the growth cone, right? And so that's kind of part of how these things move. In keratinocytes, you'll only ever see actin around the periphery of the cell, around where the plasma membrane is. Because these two cells have very, very different roles. Neurons are involved in the transmission of information, essentially, over long distances, or even short distances, but some kind of information processing. Keratinocytes are very much a structural, protective type cell. However, these two cell types, however different they look, have exactly the same genetic information. So there is no difference in terms of the information in DNA between these two cell types, or even between the cell types and organs and tissues that you see in this developing embryo. Right? However, they are clearly very, very different. Right? The neurons have a very different morphology. They're expressed in different, they're present in different places. You know, the heart has a very different shape, right? Which is a consequence of different parts of its own development, right? Things are very, very different. And you can just see that in yourself, right? You know, even stuff you can observe on the outside. There are clearly many different cell types that make us, you know, the organism that we are. And so how do we square that with all of those different cell types having the same information, right? It comes down to how is that information used, right? What things make these cells different? And that really comes down to what genes are expressed in these cells. So that's somewhat of a hierarchical issue, right? So these neurons are neurons because they were specified as such by cells around them, which had an identity because of where they are in the, even in the actual original blastocyst. But also once they are specified and they become neurons, these neurons are going to be expressing a very different subset of genes compared to keratinocytes. they're going to be expressed in neurotransmitters, depending on which type of neuron they are. They're going to be, there's an awful lot more proteins involved in transport, right? These things are long. If you think about, I'm kind of flexing my toes right now. I'm not going to show you because, you can flex your own toes, right? And I don't think Robert likes feet very much, or at least my feet. Thanks, Robert. You can do that because the neurons that innervate the muscles in your toes, or at least in like your toe bones, I guess, attached to your toes. Stop doing that. So, this stuff goes like online on YouTube. At least like the 3 people that watch those YouTube videos. Right. But you're able to do that by signals being transmitted from kind of nerve clusters basically just above your butt. Right. So there are nerve cells or cell bodies in the base of your spine that are responsible for you feeling when you stubbed a toe, and for I don't know, I guess England footballers kicking the ball in the wrong direction and screwing up their chances at Euro Cup win. Not that I'm bitter about that or anything. But anyway, right? So their role and their requirements are very different. And so this is all about differential gene expression. Some cells you'll only ever find expressed in neurons, others you'll only ever find expressed in keratinocytes. Some you'll find expressed in both because they're both required in whatever cell type you have, right? And so part of how that differential gene regulation is controlled is through essentially different chromatin configurations in those cells that shut down all the neuron type stuff in keratinocytes and all the keratinocyte stuff in neurons, right? So you don't have those genes in a position to be expressed anyway. And so there's a lot to chromatin. It's actually one of the kind of more complex and crazy ways of thinking about gene expression of all the ones we're going to learn. And I can't remember who it was. I think it might have been Adam. I'm not sure, who asked about epigenetics. Like, is it a real thing? It's very, very much a real thing, right? And it's extraordinarily complex. It's probably the kind of long and short of it. And really, it's all about modifying proteins, in particular the proteins that make up chromatin, histones, and also modifying DNA, right? So this isn't changing the sequence of DNA. It's changing the tags on DNA, right? And so all of this together is kind of comes together under the topic of epigenetics. Pardon me. And so these are modifications that can be made that are heritable, right? So you inherited some of these modifications from your parents, but don't involve the inheritance of a DNA sequence, right? So that's a real funky thing to kind of talk about. And one of my favorite kind of like wowzer sort of subjects is particularly for the ladies in the class, right? So essentially these epigenetic modifications can be made in response to your environment. So if you're in a very stressful environment, I don't know, say like working at A&MSA as a professor or in a war zone, you will actually change your epigenetic modifications. or if you're under kind of stress conditions and somehow, you know, dietary stress, food shortages, things like that. Now, if you're a guy, that's like, whatever, right? If you're a woman and you have, you're pregnant, right? Essentially, you'll be programming the DNA of your daughter in response to those conditions. Essentially, so that your daughter, when she's born, and this, you know, you change your son as well, right? But it's more interesting for daughters. When your child is born, they have kind of a bit of a look ahead to what the conditions are outside that they're being born into, right? So this is like a transgenerational sharing of information that isn't necessarily in your DNA sequence. Now, for the coolest part of this, women make their oocytes, their eggs, while they are in their mother's uterus, right? So if you think about it this way, if you're a woman or, you know, female, then the next generation that you contain in terms of oocytes is being made while you're in your mother. So you have 3 generations at once in the same body. So what the grandmother experiences, right, can be passed down to the daughter and then the future granddaughter, all in the same body. Isn't that wild? I think that's pretty cool. And that's all through epigenetic modifications. And they've actually done a couple of studies. One, I think it was in Norway or Sweden on smoking. And the other one was on a period in World War II called the Hunger Winter, which was in Holland, where the Germans essentially placed the region under blockade for helping the Allies. And so their dietary intake went down to, I think, something like 600 calories a day per person. And so they've actually followed the individuals who are in utero, like, so they're actually inside their mothers during that period of starvation, and then looked at their children, and they can actually find that there's a increase in kind of like cardiovascular disease and other kind of metabolic related issues that was tied to their parents being inside their parents during a famine. Yeah, which is just nuts, right? So the DNA doesn't change. Well, I mean, the modifications of it maybe. It's changing the proteins that DNA is wrapped around and adding tags to the DNA, right? So it's just really cool to think about. It's really cool to think about from anyone that's gone from, you know, kind of one country to another as a child, right? You know, so if you were born in one country with its own set of conditions and you move to another one, there's going to be a mismatch potentially. You know, if you were born in Sudan and then you come to the US, you know, there's going to be a large mismatch between, you know, food availability there versus food availability here. So it's just interesting to think about. Yeah, genes are cool. Genetics is awesome. That's why I'm a geneticist. And so that's kind of like the big picture stuff, but we need to look at the actual details, like how does this actually work, right? And so there's essentially 3 different things going on. We can change how the chromatin is wrapped around DNA. We can shuffle and remove histones to make some parts of the DNA more available than others at different times. We can actually change the characteristics of histone proteins, like their properties. And we can also do the same to DNA, again, without changing its sequence. And so when we come to kind of chromatin, essentially the default state of chromatin is transcriptional repression. So remember, we talked about most eukaryotic genes are positively regulated, so they're typically off, and they're told to turn on. Chromatin is part of that, right? So the default, we think, is off in terms of chromatin, or closed, right? So essentially, how that works is that we have bits of DNA that have to be available to other proteins. Transcriptional activated protein binding sites, Tata box binding protein binding sites, all that kind of stuff. And so if that DNA is physically unavailable, because it's wrapped around histones or it's kind of coiled up within a higher order structure, you won't get transcription. So we need to make those bits of DNA available one way or another if we want to turn that gene on. And so one of the main ways that's carried out is through what are called chromatin remodeling complexes, or CRCs. And they basically use energy in the form of ATP to either kind of rotate the histones, right, so that the DNA becomes more available. We can move those histones up and down DNA, and we can even just remove them entirely, right? And so, for example, here's a good one, right? We have our different protein binding sites currently kind of wrapped around histone octamers, right, in nucleosomes, so they're not available. Now, with the aid of a chromatin remodeling complex, now we can rotate those histones, and basically in doing so, we rotate the DNA that we need off of the histone, and so it's now available between those nucleosomes. So if you remember, there's always a bit of DNA, like 50 base pairs or thereabouts, between nucleosome core particles. So now that DNA is now available, right? It's accessible to other proteins. Those proteins can now recognize it, bring in RNA polymerase. RNA polymerase just punts those histones off of the DNA, and the gene gets transcribed. So from all of this about histones and proteins, you really just have to keep into mind availability of DNA. If DNA is tightly wound around histones, it cannot be accessed by other proteins. So if you want to turn a gene on, you have to somehow get that DNA off of the histone. That's really what we're talking about in terms of epigenetic modifications of histones, or chromatin in general. DNA modification is a little bit different, but we'll get to that in a second. So we can rotate them. We can simply remove them, right, by displacing an octamer. And so now we have more free DNA, essentially, because now we have a few hundred base pairs that's available. And you can just slide, you know, octamers up and down the DNA too. So you can kind of, like an abacus, you can kind of bunch them up either side of the region of DNA you need to be available. But one of the most interesting aspects of all of this is really about what we call the histone code. Right, and so the histone code is a collection of modifications to histones that essentially change it from either a closed to an open form or vice versa. So again, this is information that is not DNA sequence. This is information that you can potentially inherit that is not DNA sequence. So you can put two twins in different environments, and you'll end up with subtly different patterns of gene expression, even though they have exactly the same DNA. Oh yeah, if we have time on Tuesday, we'll definitely talk about stuff like that, Adam. That's not really what we're talking about here. That's more talking about actually changing the DNA sequence of an individual to, prevent disease in some way. But yeah, fingers crossed we'll talk about that because that's cool stuff. And so one of the key ways in which we can do that is if you look at this diagram here, right? So this is like a nucleosome core particle. We've got 8 histone proteins, got about 150 base pair of DNA wrapped around them, but we have what are called the tails. Right, these are parts of the histone protein that stick outside. They're available, they're kind of able to be modified. And so histone modifications really play on those tails. That's what gets changed. And there are 2 main changes that we care about. One is acetylation. Right. And that's typically to lysine residues. And 2 is methylation of tails and in methyl groups. There you go. Acetylation is pro-transcription. Methylation is anti-transcription. I think lysines are methylated. I'd have to double check on that. I know for sure that they're acetylated. No, not. Sorry, I've got to put the pup on the couch. Come on, then. Come on. There you go. And what this is all about is changing the charge of histones. Pardon me. Or histonins. There. And so if you remember, DNA has a negative charge, right? So the phosphate group in the phosphate sugar backbone, which you can see here, and obviously there's another one down here too, has a negative charge. And so lysines are positively charged. So normally without any modifications done to a histone, basically histone tails, in the absence of any modifications, will bind tightly to DNA. So that DNA is tightly wrapped around, tightly bound to those histones. And so it's very hard for other proteins, RNA polymerase, title box binding protein, transcription factors, anything, to actually access that DNA. It's very much kind of like, meh, mine, right? However, when those lysines are acetylated, if I can spell that, they are now neutral, no charge, and don't bind DNA tightly. So the DNA is still wrapped around those histones, right? It hasn't just kind of flowed off. But now it's kind of all like relaxed and kind of basically, you know, expands out a little bit. And that allows those proteins access to that DNA. So transcription is more likely. So now you can see how acetylation is pro-transcription. Whereas methylation typically does I don't know if it changes the charge, I don't think so. But what it does is it prevents acetylation of those tails. So if your histone tails are methylated, typically the DNA is tightly wrapped. That DNA is not accessible for transcription. If those methyl groups are removed and acetyl groups are added, now that DNA is kind of more relaxed, the structure is more open, transcription is more likely. And it's a dynamic process, right? It kind of goes back from one to the other, depending on whether or not that region needs to be transcribed or not. So in addition to modifications of the histones and their interaction with DNA, you can actually also modify DNA itself. Where are we at? And this is by methylating cytosines. So these cytosines exist that's spell that right. Groups called CPG islands. Basically what happens is that adding a methyl group to a cytosine changes what it looks like. Right? And so those proteins that bind DNA are looking for a particular shape in order to be able to recognize a piece of DNA and bind to it for whatever reason, whatever they're binding to DNA for. And so when you add a methyl group to those cytosines, that shape changes and that bit of DNA now doesn't look right. It doesn't look the same. And so that interferes with transcription factors binding. It interferes with recruitment of repressors. It interferes with RNA polymerase binding. So DNA methylation, similar to histone methylation actually, is anti-transcription. And we'll talk more about that on Tuesday. Because now is a good time to stop for lunch. So while we have lunch, if you don't have Eugene already on your computer, I'd strongly recommend you start downloading it, and maybe if we're lucky, we'll be able to install it before we start going through lab, because we're going to be using Eugene a lot today. All right.
So I actually tried installing Eugene on my computer, but it popped up as like a whole big file, but there wasn't like a exact designated installer for it. So
Right.
I was a bit lost on how to install it. Because I clicked on
Are you
the
using a
download
Mac or a PC?
HP
Or
or sorry, PC.
PC.
Yeah, because I downloaded the versions that it had, and then it went straight to a file, and I tried to look for the installer application of it, and then I didn't see anything in there. And I believe someone in the chat said they had the problem
Right.
too.
So all I did from what I can remember is click on this. It's a huge chunky ass program, but if you're willing to wait, we'll see what it looks like. See if I can recapitulate. I installed this years ago, so I can't actually remember what it was like when I did it myself. It'll be here eventually, promise.
Yeah, it pops up as a zip file,
Yeah, so you need to extract that.
and I had to hit the extract button, but I still didn't see like the official designated area.
No worries. Come on, it's getting there. Do. All right, extract all of that. I'm just going to go to downloads because I just get rid of my downloads periodically. I don't have a super fast computer, so it takes a little while. Doo-doo. It's like washing paint dry, isn't it? Where do I actually have Eugene on here? It's not there. You can actually find the yeah, I don't know. This is taking its sweet time to do. I'm not sure if it will do anything useful in the time that we have before lab starts. At least it's doing something. So why don't you at least go through the same process that I am to make sure you have those files extracted somewhere. And then by the time we come back from lunch, hopefully this will have finished. And I'll be able to figure out which one it is that you should be working with. All right, so because otherwise we're just going to sit here watching my computer do something for 25 minutes instead of eating some lunch, which would be no fun at all. So yeah, try doing the same if you haven't done already. And hopefully by the time we come back, this will have done something useful and we'll be able to use that information. Cool. All right, everybody. See you at probably a little bit past 2, probably like 2:05, something like that. Takes me a little while to eat my lunch and stretch my legs. And yeah, and then we'll go through the Eugene part of the handout that we started on Tuesday. And also, don't forget, we've got another lab potentially next Tuesday if we need it, in case you have any problems today. So don't stress too much. All right, I'll see you in about half hour. Bye.